De todos modos. Anyway, you can forget all that rubbish. And now I'm going to talk about something very, very different. Just give you a big overview of what we're doing with a large cohort, a population cohort that anybody can start. And it's a quick tour of what I'm doing. Oh dear. Have I done something wrong? Should I try this? Puedo ver el ordenador que funciona bien para mostrar. Okay. Okay, I've got a question. Hands up if you know how many genes there are in the average human. Simple question. Give or take, you know? You don't have to know the exact number. How many human genes are there? Just, you don't have to give us the answer. I just want to work out how many people. So, is it around 20,000? Is it around 200,000? Is it around 2 million? 23 million? 230 million? How many people don't know the answer? I'm going to ask you if you don't. Okay. Okay, the answer is 20,000. The average doctor in the UK, only about 1 in 5 know the answer. The average cab driver in London is about 1 in 3 knows the answer. Okay? So if you want to learn any genetics, you ask a black cab driver. That's the rule. The other point I'm trying to make here is that the average worm, many species of worm, have more genes than the average human. And we like to think we're a bit more sophisticated than the average worm, which suggests that there's more to life than just the number of genes. It's what you do with them. Okay, let's see if this works. Yes. Okay. Making progress. So, as I told you earlier, I've been studying these twins for 20-odd years, 11,000 of them, most are female, and we've done all kinds of tests on them. So including GWAS, methylation, whole genome sequencing, gene expression, we have longitudinal DNA, we look at environmental pollutants, we look at immunophenotypes, metabolomics, glycomics, telomeres, microbiome, and metagenomes. We also have 3,000 clinical phenotypes, and we have several hundred international collaborators. So you put all this together, we're not short of data. And if anyone wants to play with our data, you're very welcome to collaborate. Now, twins are the ideal perfect experiment. They've been called the equivalent of the mouse experiment in humans, because nature has given us this these two types, identical twins who share all their genes with non-identical twins who share half their genes. By comparing the two, you can work out what is nature and what is nurture. And this is what, for the first 10 years of the registry, we did, because the genetics wasn't so good. So what we did is working out what things were heritable and what were not heritable. How many people here know what heritability means? Is it how much money you get from your parents? No. It's not what the risk is of getting a disease. It's not the risk of passing it on to the next person. It is the proportion of any trait that is due to genetic factors. So in this room, there are some of you that will fall asleep very quickly in seminars, even with this Arctic air conditioning. Others will be wide awake even after 4 hours on a very hot room. That's the variation. And on average, I could say that the heritability in this room of 60% for falling asleep in seminars would be about right. But it might be very different in another room with a different population. It's very population specific. So heritability is actually quite a specific term. So when you're looking here at this group of traits, you can see it goes all the way from 30%, which interestingly is longevity, which is one of the lowest ones, all the way up to 90% for corneal thickness, and for those of you who can't see it, the third one down, myopia, which is highly heritable, and that's a joke. Now, the average heritability of most diseases is about 60%. So if anyone asks you about your favorite disease and you don't know the answer, just say about 60%, and you'll usually be correct. Now, when I first presented these results, many people got upset because they might have been sociologists who worried about IQ being heritable, intelligence quotient. Or the other people said, if you say something is highly heritable, that means we can't change anything. That's terrible. It's a really bad message. You're giving people the wrong message. Well, myopia is 90% heritable, but in many countries of the world, like in Singapore and other Asian countries like this, it has tripled in the last 40 years. So that now you only find one kid out of 1,000 aged 18 who doesn't wear glasses. And that's with a heritability of 90%. And if you do the same study of the same Asian kids in Australia, where they play outdoors, the rates are much lower. So a small threshold change in environment can make a big change to a condition, even if it's highly genetic. So do not be frightened if someone says it's highly genetic. They can still be a public health message. Same is obviously true of obesity, where we're going through a massive epidemic. And in all countries, even in super healthy Panama with its some of the US habits on fast foods, just because it's genetic doesn't mean you can't do something about it. Okay, now what about some other traits? So most of the ones here are not a surprise, but you might be surprised that back pain is 3 times more heritable than breast cancer. So some things are surprising. Okay. Now what about other traits? So-called environmental traits that the epidemiologist said is mine, keep away genetics. Physical activity is about 70% heritable. So whether you like to sit and watch TV or actually like to do the sport is largely due to your genes, as is how much beer you drink during the game. Whether you like junk food or salads is also heritable. Whether you smoke a lot is also heritable. Your food preferences, whether you like garlic, spicy chilies, or kimchi, if you like Korean food, is also heritable. Whether you get bitten by mosquitoes, which many of you are interested in, or in the other room they're discussing about vaccines against some of these nasty new viruses spread by mosquitoes. Some people never get bitten by mosquitoes, others do. Highly heritable because of the different chemicals on the skin. Voting Conservative or Socialist. In the UK, and I was brought here by the British Council, so I can't be rude about the government. It's a great government. And David Cameron, very popular leader, voting for him, he got 60% heritable figures. So whether you like him or not, a lot of that is due to your genes, not just to whether you like his pretty face. Whether you're extrovert or introvert is also, your personality is also heritable, as is your sense of humor or not. And lastly, the one that people find most difficult to believe is your belief in God. It turns out that whether you, not whether you go to church, but how strongly you believe in God has a genetic basis based on at least 3 countries where we've done twin studies. Which also means that your beliefs, whether you're a skeptical person or a believing person, also means whether you believe my talk or not is strongly genetic. Moving on to genome-wide association scans, which we used the twins for, and very successfully in many studies. These use genetic markers, up to about 1 million genetic markers, which you can now get done in volume for about $30 or $40. If 10 years ago you'd said you could get this test done on 1 million markers for $40, most people would not have believed you. But you can now, and it's been a very successful business, finding hundreds and thousands of genes for hundreds of different diseases. What it hasn't done is predict. It hasn't been able to predict diseases, which is what we thought it would do. We thought we'd get these genes, we use them in prediction. I can tell whether you're going to get diabetes or you're going to get Alzheimer's or whatever, and it turns out that was totally the wrong thing. These are very non-predictive, but they are proving very useful for mechanisms. Understanding the genes, these are robust results that are replicated hundreds of times and are very useful in many other research areas. And the mechanism itself, by changing it, you may be able to get a much greater effect on the body with a drug. But in terms of prediction, they're useless. And we'll come back to this later on. But the twins, just our twin sample has been involved in 9% of all the GWASs performed in the world. So it shows if you get a good cohort with many phenotypes, everyone wants to be your friend. So that's, in general, disease GWASs, which haven't been useful for prediction, mainly because it's too far away from the gene. Too many complicated things make up a single disease. But if you take the next level down, the intermediate phenotypes that go under a disease, such as metabolomics, such as immunomics, glycomics, proteomics, etc., these are associated with disease, and they're also associated with genes, and by looking at these, you might get much closer to the action and mechanisms. And an example of this is if you do a GWAS study for, say, rheumatoid arthritis, and you get these hits, golpes, and you find out that usually they're not in a gene at all, so they're not giving you information, but you know they're reliable. You correlate those SNP hits against these other intermediate traits like immune traits. You can work out what's going on and understand some of these mechanisms. And to give you some more examples, we're now able to look at just from 15 mils of blood, get 78,000 white cell markers. Now, when I started this study, I thought there were only 10 white cell markers, so this was a bit of a surprise to me. It expanded my knowledge to realize there was such diversity in our white cells. It turns out that some of these are co-correlated, so there are perhaps only 30,000 independent white cell markers, but that's still quite a lot for most of us. And when you look at the heritability of these 30,000, you see different patterns emerging. Many of them have a strong genetic component. Some of them have no genetic component. And also, just the way the surface protein is expressed is also another way. So not just the amount of the cells, but actually what's the protein that's actually stuck on the cell also has a genetic component to it. So it turns out that about 2/3 of our whole immune system has a strong genetic basis. And you can look at these subsets of these. The immune system, which is important, and it turns out that the innate immune system is much more environmental compared to the other components. So the more sophisticated it is, the more genetic, the more we've evolved as humans to have a really sophisticated, specific immune system. But as well as understanding immune genetics, it also helps us get close to finding genes for these. And so we published this work recently just as an example that just in 400 people we did a study where we looked at these immune cells to try and find genes, and we found hundreds of genes in 400 people. My first GWAS study, we looked at 4,000 people and found one hit with a conventional phenotype like bone density. So by picking the right phenotypes, you get much more bang for your buck. And there are going to be many more of these studies, and we're continuing to do many more of these. And I think particularly in areas such as autoimmune disease or infectious disease, or in why people respond differently to vaccines, these could be absolutely crucial. Another favorite omic of mine is looking at blood metabolites. And you can now, just on 0.5 ml of blood, measure 1,000 metabolites very accurately for about $250. And it's the same principle as I showed you. These are very close to the genes. These are the circulating metabolites, but as well as the traditional ones, for example, underlying glucose metabolism, etc. You've got amino acids. You also can measure caffeine levels, whether someone's had a coffee recently or they've taken an ibuprofen recently. Extremely specific. You can tell whether they're a fish eater or they like drinking red wine. These are things that are going to become more common as we abandon traditional testing and we go towards these exciting new ways of looking at the human body. And there are some hospitals in the US treating pediatrics where they screen all the unusual syndromes in children with this metabolomics in order to more closely pick up and more quickly pick up metabolic disorders rather than the genes. The genes can take many months or years to actually work out what's going on, but if you see a metabolite that is 100-fold higher, you know instantly what to do about it. And again, we've combined this metabolomics with our genetics to understand what's going on, and it's been extremely useful in building up maps like this one here, where you have networks of these metabolites with networks of genes. And for people designing drugs, these are extremely useful. And we did this study with Pfizer, and they're still using this network in their design of new drugs to see where they can piece together, by knowing that they know some of the genes, they know some of the metabolites, guessing what this drug would do. And these are increasingly being used for these large databases. Now, proteomics is another omic that we're just getting into. The problem with proteomics is we don't know how many proteins there are, which makes it kind of tricky. We know there are at least 5 times as many proteins as there are genes, but there could be 20 times. It depends how you define it, and we haven't got there yet. But increasingly, there are methods now that look as if they're ready to be used, and we've had a look at a couple of these using mass spec and immune capture methods. And mass spectrometry is looking good, although it can pick up many of them, but can be hard to sort out computationally. And we find heritable signals, which is, again, a good sign that we're doing the right thing. We've looked at various immune capture methods, and this is one that just looks at like candidate proteins that everyone's interested in. So everyone's favorite protein is in this list of 1,300, and they can be measured pretty accurately. And again, we find that these are nicely heritable, and they also correlate with age. And to make a good biomarker, we find they have to have a range of heritability, so some are heritable, some are environmental, but also they're pretty good at predicting age. And metabolomics is good at predicting age, and proteomics is also very good at predicting age. And if you just took the top 11 proteins, you can get a pretty good guess about somebody's chronological age. And these compare very well to other methods. So I think we're going to see more proteomics in the future. It's still a bit expensive, but obviously the protein is where the action is. And so the basis of most therapeutics is in this area, and therefore I think this is coming. How many people here have heard of glycans? Okay, about 10 of you. Well, I hadn't heard of them for 20 years until someone reminded me, and it's making a renaissance now. Most people haven't heard of them because they're so complicated to explain that most people give up, because they're the very complex post-translational modification of sugars. These are of proteins by sugars. So as a protein is produced, it's then modified further by these sugars which attach itself in lots of different ways. And the model is this immunoglobulin, IgG. And slight changes can make a big change to the function. And if you look at this schematic of how the immune system works, you've got the antibodies sticking onto the antigen, and of course you've got the role of all these other things that come off it. So the precise way in which these antibodies are configured is really crucial to our survival. So knowing there's this whole other layer in there that's helping this on its way is important. And we know that the glycan can modify this so that each person in a way gets its own personal barcode. We all have our own barcode, and this changes. And we'll now be able to measure this quite easily in blood, fairly cheaply for about $100, and you can get 80, everyone gets, just for IgG, we all have 80 different subtypes. And what's interesting is it's extremely correlated with age. And so just by measuring 5 of these, you can predict age better than many commercial tools, like telomeres you might have heard of. The correlation with age is at least 2 or 3 times better than with telomeres. And so clearly our immune system is very much related to our age, and this is perhaps one clue about why that is. So you'll be hearing more about these glycans, and also the genes underlying these glycans also tend to be those implicated in immune diseases. So it turns out we have genes which may be acting on our causing disease by modifying these glycans. This could also be the reason why people respond differently to vaccines, because many of the genes underlying these are related to response to vaccines. Now, coming back to the twins, which I left 10 years before, I used to study just why twins were so similar. And when you look at a picture like this, everyone is drawn to how similar identical twins are. You look at their eyes, you look at their expressions, you look at the way they smile or they hold a cup of tea, and you're struck by that. But you're being fooled, because the human brain looks for similarities much more than it looks for differences. And when you look at these people in detail, you see many differences between identical twins that you weren't there at first sight. And there are certain identical twins who really do look quite different. And when you started to look at the literature, and I started to do this a few years ago, I found that twins rarely died of the same diseases. In fact, they rarely died within a few years of each other. And that's why the longevity heritability I showed you was quite so low, which is not what you would expect at all. And when you look at the concordance rates on this chart, you can see that for most of the conditions, it's below 50% similarity. So that if one twin has one of these conditions, the chance of the other twin having it is much less than you would expect, because most people would think it would be about 90%. But just take rheumatoid arthritis. Affects 1 to 2% of the population. If you had an identical twin with it, your chance of getting it is only 1 in 6. Very low. These are identical clones brought up together. So what's explaining this? What are the differences between these people that make them quite so different? And a few years ago, I thought the reason must be epigenetics. The genes are the same, the structure is the same, but if the genes can be switched on or off by epigenetic mechanisms, that could be the reason. And it turns out that identical twins have different epigenetics. And so it could explain it. And so we got this large grant to measure the methylation changes in thousands of the twins across a whole range of diseases to see if we could find the differences. The basic model was we were looking at pairs of identical twins where one had a disease and the other was normal. And we did this many times to try and see, this should be the perfect design to try and find small differences between people where you adjusted totally for genetics and family. It turned out it was a lot harder than we'd thought. And we did find changes. We've published on some of these. We found differences in a pain receptor, in pain sensitivity. We've found differences for depression in one gene. And we found some differences for breast cancer. But many of these we've found no major changes. An example here is type 2 diabetes, which we were expecting rather big changes, and it turned out we only found one gene that was significantly altered methylation-wise, and that turned out to be a consequence of the disease, not a cause, because we had expression data to show that it wasn't affecting expression. And the reason is that these epigenetic changes are very small and very subtle. We thought they'd be huge, but it turns out we're looking at very small percentage differences in methylation. And so with the current tools we haven't been able to get nearly as many good results as we'd like. So we're really waiting for some new technology here. But technology is good in another area of expression. So expression with arrays, which we've heard about in the last talk, and also an RNA sequencing is now extremely precise and getting cheaper all the time. And whereas I don't think it's useful in prediction, again, it's telling us a lot about mechanisms. And we can look at this sort of data and see many of these expression changes that are linked to genes and put all this stuff together to look at the differences across different tissues. So many genes are expressed equally on all tissues, and some are very specific. And this is a paper we've submitted about age and expression, showing people the differences in variance when people age. And we expected that as you get older, there'd be less genetic control. The variance would increase. everything goes to pot when you get older. All goes downhill. All the controls go out the window. It turns out that's not the case, and actually there's more genetic variance as you get older. The genetic control on expression is actually greater. So many of these assumptions we've made are starting to look different when we have this new technology. Moving on to my final and my favorite omic, the microbiome. We have 100 trillion microbial cells, which is 10 times more cells in our colon than in our whole body, if you exclude red blood cells, which don't really count because they're not proper cells, really. Most live in the dark in our large intestines, and we've forgotten about them. If you put them all together, they weigh about 2 kilos. So it's been called our forgotten organ, and it's been linked to all kinds of diseases. Now, we've been studying these in twins, and we know that when we're born pretty much sterile. And we get our microbes through the natural birth process. And it turns out that the first 3 years of life are quite crucial in developing the right microbes that then go on and form our immune cells. Because we know that in mice who are brought up germ-free, they don't develop a normal immune system, they don't develop a normal brain, and they need 25% more calories just to stay alive. So these microbes are absolutely crucial for our existence, and yet the last 40 years we've been trying to destroy them. And another thing that's destroying them is cesarean sections. I don't know the exact figures for Panama, but the countries around, it's around 50% of births are now through cesarean section. It's 24% in the UK, which is still too high, and it's 80% in Rome, and 70%, I think, in many parts of Greece, 60% in Brazil. Most of this is for cosmetic reasons, and because the obstetricians like to play golf and don't like to be called out at night. But it's a very bad trend, because a baby born by cesarean section acquires whereas in a normal birth, the baby acquires the microbiome of the mother, and that's what it's designed to do over millions of years of evolution, because the baby's head passes right next to the mucky bits of the mum, and that's how the microbes get into the baby's mouth and colonize. And they're essential for fertilizers for digesting the breast milk and getting the immune system right. By cesarean section, the baby resembles the microbes of whichever nurse picked them up in the operating theatre, and usually just their skin. So I was born by cesarean section, so I'm a bit worried. And it turns out that not only are the microbes very disturbed, but also it's related to increases risk of obesity and allergies. So this is just one reason where modern medicine is going wrong. And they also routinely get antibiotics as well. Just before the operation, mothers get antibiotics. Anyway, back to our twins. We've been studying them, and it turns out that our microbes are very different. We only share about 10 to 20% of our microbes with each other, and we didn't know whether this is due to what this reason was, whereas you compare that to our genes, where we share 99.9% of our genes. So our microbes could be one of the big reasons we differ so much in diseases, even in identical twins. And it turns out our microbes also produce about a third of all our metabolites. So they're little factories. They have 200 times more genes than we do. They're producing these enzymes, these chemicals, these vitamins, and they're extremely important for our health. We didn't know whether there was any genetic component to this at all, And we did a twin study to look at whether there was heritable component, whether the reason we're all different is because we attract different microbes, just like we attract different mosquitoes. And the simple answer is yes, there is a heritable component. It's not that strong, and it's only for certain microbes. So some microbes you get just because of your diet, others because of your genes that attract certain microbes to you. And we're still doing more work on this, but around 30%, 40% heritabilities is what we're seeing at most, not very strong. But in the same study, we also looked at obesity, looked at fat twins and thin twins. We found a few microbes that were always associated with thin people. If you were skinny, you always had this microbe called Christensenella, which no one had heard of before, and there are several microbes that are protective against obesity. If we took them from the twins and we put them into mice and we fed them junk food diets, it could keep you skinny. So this is a fat-busting microbe, as the tabloids called it, and there's a few Japanese websites selling it. You can buy it and put it on your cornflakes in the morning. Just want to tell you about a brief experiment I did. I was doing various experiments on myself and my diet because I was writing a book on this subject. And I'd just done a 3-day cheese diet, French unpasteurized cheese, which was great for the first day, okay for the second, but by the day 3 I'd really had enough. And the next experiment was to do a 10 days McDonald's challenge. But I decided I wasn't really up to it. I wanted to pick someone who was better qualified than me, who happened to be a student. He was hard up for money, and he really liked McDonald's. And he also happened to be my son. So he was delighted to do this, and he took up the challenge. He was great starting. About day 4, he'd had enough, though. He said, "Dad, can I stop now, please?" I said, "No, you've got to finish the 10 days. All your meals have got to be eaten at McDonald's, but I will give you are allowed to have chicken McNuggets instead of Big Macs sometimes." So he was happy with that, it kept him going, but nothing else. But by the end of the experiment, day 10, he'd really had enough. And his limited studying powers reduced even further. But the real change in him was not just to his skin and his mental state, but his microbes. He'd lost 40% of his gut microbes in that diversity in that time. And it turns out that the diversity of your microbes is the #1 predictor of bad health. And to get more diverse microbes, you need diverse real food, vegetables and fruits and grains. And also, the other thing that, as well as not getting any fiber in the McDonald's, apart from the small bit of gherkin, you also are getting many chemicals, emulsifiers, preservatives, which also affect the gut microbiome. So good reasons to avoid it for your microbes. Antibiotics are also a disaster. They're like a nuclear bomb each time you take them, going off in your gut. And the average American has 18 courses by the time they are 20, and I think it's pretty similar here. Average 3-year-old having 2 to 3 courses on average, with many having 3 times that. Most people can cope with 1 antibiotic course, but if you keep getting them, they will never recover, and you get very dysbiotic. And again, there's a strong association now with obesity and how much antibiotics people take. Now, if you want to get rid of your microbes, you don't like them, these could be the tablets for you. You just take 30 of these, and you'll get a new gut microbiome. Technically, they're called frozen poo tablets. The Americans call them crapsules. And they're very effective. When treated for Clostridium difficile, which kills thousands of people, these have a 90% success rate. So they are mainstream medicine now. It's not a joke. And they're currently treated in 500 centers in the US for Clostridium difficile, and they're going to be starting treating diabetes, IBS, IBD, and maybe autism in trials soon, although the data are not yet there. So watch this space. So to give you some summing up advice, if you treat your guts like you would a perfect English garden, you won't go far wrong. So plenty of diversity, changing your soil, making sure you profit from the seasons, sprinkling on probiotics, adding fertilizers, prebiotics to it, avoiding toxins like chemicals in food, avoiding antibiotics, and above all, diversity is the key. And remember that with 100 trillion microbes inside your gut, you never have to eat alone again. And finally, I'll just leave you with this slide, which we have a crowdfunding study so people can test their own microbes in Europe with the British Gut Study, and we have a parallel study in the US run out of San Diego where you guys could sign up for a small amount of money and help citizen science as well. And if you're interested in this, you can also get my book on this subject, which sadly hasn't yet been translated into Spanish. I'm very sad. But on that note, muchas gracias. Tomo las preguntas. Gracias.
Thank you, Dr. Spector, for your excellent lecture. I just wanted to comment something that was not too clear. I understood that you say that in the bowel, our bowel, we have a lot of bacteria. It's not an organ, okay, but it's like
an organ.
Heavy organ. Okay. Can you comment on autism with this bacteria?
Autism, yes. So,
have you heard something about that?
Yes. So many diseases have been studied with their association with gut bacteria, and there have been about 5 studies of autism and gut bacteria, all of them showing abnormal gut bacteria in autistic children. The problem in autism is to separate difficult eating patterns from the gut microbes. But so we know it's disordered. We don't know if it's cause or effect, or both. But there have been some animal studies where they've transplanted gut microbes from autistic children into mice and caused behavioral problems. So there's something to the story. We need to learn more. But we also know that the gut microbes can produce brain chemicals. So they can produce GABA and serotonin, which can affect the brain, and there are a number of elegant experiments to show that. So I think it's quite possible that autism is related to gut microbiome, but we haven't proven it in humans yet. But it's an exciting area, as it is in also possibly in depression and other things. But
look, thank you very much. I really enjoy your presentation. I have 2 quick questions. So, for example, for heritability, like I believe, like when you compare like the genetics and then all the environmental factors and lifestyle choices, is there like, do you believe there will be like many environmental factors that people are not like thinking about that could rule out like genetic heritability and make it like less susceptible? For example, let's say mother's diet or the exposure of like people to nature or cesarean, for example, and they could rule out how important are these genetic factors for diseases. And then I have a second question, and it's like you were talking about cesarean. I was thinking like if you have ever find a case of twins, identical twins, that one was born naturally and then the second one was born by cesarean. Like if you have seen something like that, or what have you found about that, would be interesting to know. So I hope I explain myself.
Okay, so yeah, there aren't enough cases where twins have been, I mean, I think the world record for twins born apart is something like a month apart. One twin got forgotten. So there are, but unfortunately there's not that many to study, so it's rather hard to get, and a bit like, it is more common to have one born by cesarean and one naturally, but again, we need big numbers to do these studies, and so it's difficult. Interestingly, in our twins, the average age is 60, and in that population, only 10% were born by cesarean section. Now, in London, where rates are quite low compared to the rest of the world, it's 50%. I expect in most of North and South America to be about 90%. So these individual ones, they're anecdotal ones, and usually there are other problems that make it difficult to generalize. I think the other question was, I'll rephrase it. We used to think there was environmental and genetic factors. They were quite clear-cut. Now I think they're mixed. We haven't really found, I've been struggling with hundreds of traits to find anything that is not genetic. Okay, if people can come up with any suggestions that are reasonable traits in a population that have no genetic component to it. The only one I've found so far is your favorite football team. Okay, so whether you support Manchester United or Arsenal, or it seems to not be heritable. Favorite color is another one. We didn't find anything heritable there. And also whether you like Mr. Bean or not. Whether you find him funny. Not heritable. But apart from that, I'm struggling. But I'm welcome to other examples.
So I just had two quick questions of things that I found interesting, and it was a wonderful talk. So you showed that for the twins that you've been studying, 85% of them were female. And is that because more female want to participate in your studies, or because that's the general percentage of twins?
No, it's roughly 50-50. But there's a bias towards, in England we don't pay our subjects, and so they're altruistic, and so women are much nicer people than men for science, and so they volunteer. And also they don't faint when they have blood taken with needles and things like men do. So they're much better generally. But also when I started 20-odd years ago, we specifically only collected women because we were looking at female predominant diseases related to menopause and like arthritis and osteoporosis, etc. And more recently we've tried to get men, but we're not as successful.
And the last quick question, for that study, the McDonald's study, do you need to get an IRB approval for that study?
No, and my son is still threatening to sue me, so I have to keep him supplied with more student grants and things, yeah, so. Yeah.
Tim, couple of thoughts. The not picking up the microbiome at the time of birth if you delivered by cesarean section, would you recommend a similar sort of intervention as to the one used for treating H. pylori in terms of sort of reseeding the gut?
Yes, well, I didn't mention it, but what's called inoculation is what they are currently doing, and there have been some studies in Puerto Rico, which has a very high C-section rate, where they are in C-section, they're randomizing babies born by C-section to inoculating, where they take a swab from the mother's rectum and vagina and smear the baby's face with it. And randomizing those studies, and the first pilot studies are looking very encouraging, that you can repopulate what nature intended with those methods. And several Scandinavian hospitals are already doing this, them being very trendy places, and so they're offering this to C-section women. And several of my colleagues in the microbiome world, if they've had a child, they've suggested to their partner that's what they also do. So it's becoming very fast seen as a safe, effective way to counteract the artificialness of modern medicine.
And secondly, is there anything to be learnt from studies of heterozygous twins in terms of the influence of environment over genetics?
Yes, well, we couldn't get these results without the dizygous twins anyway, because we need a comparison group. So they're the perfect comparison group to always gauge what, if you just had a common environment and shared half your genes and the same mother. So we've learnt about gene-environment interactions from that group as well. So half of our twins are non-identical and half are identical, and for most of these experiments you do need both. So they have been extremely useful.
The last question.
Hi, nice talk. So I have a question. Do you know if it's any relationship between microbiome composition and aging or age-related diseases like Alzheimer's?
Yeah, it's a good question. So we've just published last month on this. There is a correlation with frailty. So frailty is a state where it's in a way accelerated aging at the same chronological age. And we show that frail individuals who have, for example, lower muscle grip strength and differential blood levels are just aging faster than other people, have a disrupted microbiome, and they have less diversity, and they also have increased levels of inflammatory microbes. So this seems to be a common occurrence in many diseases, and the aging is you get less diversity of your microbiome and also an increase in inflammatory species. And this is a pretty good predictor. So I mentioned earlier that getting a sample of your poo, I can predict many more diseases than I can with your DNA. 10 times better, whether it's obesity, diabetes, whether you're going to die early or not. So I think we're going to learn much more from looking at our microbes, whether it's in our urine, maybe our skin as well for protection against mosquitoes or whatever it is. Extremely useful. But and predicting which frail people, old people, need extra nutrition, for example, could well be from the examining the feces of elderly people. Okay, but hopefully all your guts are all very healthy and increased. So do remember, keep diverse and you'll be fine.